Obrigado and dank u wel. Thank you for the amazing introduction and to be here. This place is amazing. Really, the scenery, Lisbon, I mean, it's really a pleasure to be here. So, and thank you for the really incredible introduction. I hope it's recorded so I can show my kids. They would, so I'm in New York, as was mentioned. And this is, I show this because this is a satellite photo of Manhattan. And you know, to start off the conference, I think a lot of what I'm going to talk about is this idea of neighborhoods and spatial. And like any big city, you know, New York is a neighborhood, or sorry, a city of neighborhoods. This is where I live on the Upper West Side. This is the Upper East Side. And sometimes those neighborhoods encroach on each other. Sometimes they're completely isolated. And sometimes one neighborhood will turn into another neighborhood. And I think this is what happens all the time in cancer. And I'm going to tell you about some of our work there. This is probably a slide or a schematic that we'll see presented several times at this meeting. It's a very important one. So in terms of the immunotherapy response to cancer, after tumor mutational burden, probably the single most important factor as to whether a patient's tumor will respond is how infiltrated that tumor is. So there's at least 3 different archetypes of tumors. The kind of 2 most famous are the deserted or excluded, in which the tumor core doesn't have T cells in it, and it's usually surrounded by a layer of macrophages. And then there's the inflamed tumor, and that's when the T cells are actually in and found throughout the tumor. And in this type of spatial organization, these respond much better to immunotherapy, and these respond the worst. And then if you have an infiltrated tumor that has these tertiary lymphoid structures, which are these kind of almost like germinal cell zones in the tumor, they tend to respond even better. And I show this picture. This is from a paper published by my colleague Sasha Nyadik. It's a melanoma, and I just think it's so striking, this pattern. And you know, here in the middle, it's the cancer cells surrounded by a layer of macrophages, surrounded by a layer of T cells and B cells. And when you look at that, you immediately realize, you know, if I'm going to inject CAR T cells or I'm going to give anti-PD-1, if these T cells can't get into the tumor core, none of those therapies are going to work. And that's why this type of tumor responds poorly and this type of tumor responds better. The other thing to think about when looking at this, which is really interesting, is that even if that melanoma metastasizes to a different location, it ends up taking on the same spatial architecture, telling us that there's some sort of programming going on which is creating this type of patterning. And I'm just going to take this to a different place other than cancer, because I think that too often we forget about this very interesting phenomenon, which is that in autoimmune disease, or all T-cell autoimmune diseases are spatially localized. And this is an absolute mystery, but it has lessons that are critical for our understanding of tumor biology. So this is vitiligo, alopecia areata, and type 1 diabetes. And this one, we often don't think of it this way, but in these 3 types of autoimmune diseases, you have this phenomenon where a T-cell has killed a melanocyte here, but it hasn't killed it just a few microns over. Okay? Same antigen, same target cell type, but something is causing or allowing the T cell to kill this melanocyte and not this one. And the same thing here. This is a pancreas. Some beta cells are killed, some aren't. Why is that? And this phenomenon has incredibly important lessons for cancer, because we tend to think that if a tumor is out of neoantigens, we're done. But this right here, phenomenon that happens in more than 5% of the population, tells us that we can take a self-antigen and turn it into a targetable antigen from the immune system in a localized way. And you can do that if you change the tumor microenvironment. Okay? So then the other thing to start the meeting is this very important concept that, at least for those of us who do a lot of mouse modeling, we don't often think about, but as soon as you go to the patient, you realize that really probably the most important factor in how a patient will respond is how heterogeneous their tumors are, because that is the source of resistance. And that happens all the time. So you can have pre-existing resistance, or you can have adaptive resistance, but it's a key problem. And this is a really nice work from Nick Navin showing you how These are spatially mapping tumor clones in a ductal carcinoma in situ. And what you can see here, green, red, and blue, those are 3 different cancer clones. They're spatially organized as clusters in the patient. This is not a mouse model. And when they did RNA-seq from laser capture on these, what they saw is actually, even when clones were neighboring each other, they had different tumor microenvironments. So you can see that you have green and blue clones almost side by side, but the blue clone has an interferon alpha and interferon gamma response, and the green clone doesn't. And so you have these different neighborhoods within a tumor, spatially separated and dependent on the underlying clone. So my lab has been trying to study what are the factors what are the genes that drive this organization. So I said before, the melanoma can recreate this, and many tumors can, wherever they go. That tells us there's something that the tumor is doing to program that microenvironment. And then we're trying to do something that doesn't often get done when we do a lot of modeling work, which is factor in heterogeneity to understand how clones compete with each other. So my lab does do a lot of functional genomics. We've been doing this for about 15 years now. We started with microRNA and shRNA. And we were doing a lot of these CRISPR screens, which we love a lot. It's a beautiful technology. Take a lentiviral vector library, you drop it onto cells, cancer cells, you transduce them, then you apply some selective pressure, time, T cells, drug. You extract the DNA, you throw it on a sequencer, and you ask the question, what is my frequency of a guide RNA or a gene knockout before or after I apply the selective pressure? It's an amazing technology. It's very, very powerful, but it's really limited to looking at cell intrinsic functions. So whenever you see a CRISPR screen, there's 2 things that you should think about, is that in general, what they're looking for is something that is cell autonomous, like, does my cells, do they proliferate more? Do they die less? Do they express marker X? And so you're not accessing with any of these CRISPR screening technologies what's happening outside the cell. Do I have more infiltration? What does the macrophage look like? Where is my cancer cell located? And the other thing that people don't often think about in these CRISPR screens is that they're not actually assaying for genes that are operating extracellularly. So not only are they not assaying for functions outside the cell, they're not assaying for those genes that work outside the cell. So what do I mean by that? Well, if you have a Petri dish and you've knocked out any one of a cytokine, IL-6, IL-12, and it's only knocked out in 0.1% of the cells, the other 99.9% of the cells are going to keep producing that factor, and so you won't actually find out what that gene does. So a couple of years ago, we came up with a technology. I'm going to skip through the details. It's a protein barcode technology that allows us to read out CRISPR screens at the protein level. We call them procodes. And all you need to know is that each procode is linked to a different guide RNA. You can buy them on Adgene. I get no money from this, but it's a technology that's very easy to access. And we published a few years ago. But what we've been doing with it for the past few years is using it to perform CRISPR screens that we can read out spatially. So what we do is we take cancer cells, we put a CRISPR library on where each CRISPR is linked to a different protein barcode. We implant the tumors. We almost always do it orthotopic models. And then we pull out the tumors, and we can actually determine which guide RNA and therefore which knockout a clone is carrying, depending on which procode they're expressing. And I'm going to show you we can also pair this with spatial transcriptomics. So this is an example. Many of you may have seen this before. We took KP lung cancer cells, we put in a procode CRISPR library, and we inject mice. This is 120 different CRISPR knockouts. You can see that you have tumors all throughout. This is the H&E staining. And now when we do multiplex imaging for the protein barcodes, you can now see here, we can resolve which knockout or which procode and therefore which knockout each clone has. And in this particular model, what you can see very clearly is that each of these tumors was started by a single cell that grew out. And in some cases, you can actually see a nice mix, and this becomes important for what I'm going to show you later. So this is what it looks like. We can now read that out. There's one lobe of a lung, and you can see that each of these has now been labeled with a particular gene knockout that they're carrying. And now whatever phenotype you can stain for, you can analyze in what we call perturb map. So this is an example where we put a library of 35 different guides into this mouse. This is all throughout the lungs, and we see these different genes come up. Since this is published, I'm going to try and skip through it quickly, but I just want to focus on these two genes here, SOX1 and TGF-beta receptor 2. So when we knocked them out, those two tumors grew the best. They were highly enriched, telling us that knockout of these genes gives these tumors a fitness advantage. But we don't know why that is. And so what we're able to do is we stain that tissue. We actually stain all the different tissues. This is very cheap and easy work. Anyone who can do imaging can do this. We stain for these different markers, B220 for B cells, T cells, myeloid cells, and then epithelial marker. And the radial plots, if there's a bar on the outside, that tells you there's an infiltration in that tumor lesion of those particular cell types. And if you see the bar on the inside, that tells you there's a depletion. So in one experiment with 10 mice, we can determine how 35 different genes influences the tumor microenvironment very rapidly. And so that's what you're seeing here. Each of these plots is telling you what the TME is of each of these knockouts. And the one I want to highlight here is the SOX1 and TGF-beta receptor 2. So those tumors grew better, but they had opposite TMEs. When you knocked out SOX1, you had a T-cell enriched tumor microenvironment, and then when you knocked out TGF-beta, you had a cold tumor microenvironment with fewer T cells in them, and yet these were both highly enriched. So we asked a question that often doesn't get asked, and that is, how does one neighborhood affect another neighborhood? We wanted to know if you had a hot tumor beside a cold tumor, what would happen? And me as an immunologist thinks, especially thinking about T cells, I think, well, immune cells are great at crawling and moving wherever they want. But just like I showed you in vitiligo and in diabetes, that's not always the case. And so what you see here is, I think, really remarkable. In white are the SOX1 knockout tumors, and in blue is the TGF-beta receptor 2 knockout tumor. This is an example. We had almost 2,000 different tumors here, but this is an example where the two tumors are side by side. And in red is the T cells, and in green, or green and red are the T cells. And you can see right here, that black hole is exactly where the TGF-beta receptor 2 knockout tumor is. So almost to the cell, you can see that that tumor, even when it's surrounded by a hyperinflamed tumor, the TGF-beta receptor 2 knockout tumor is not getting infiltrated. It's maintaining its neighborhood dependent on that underlying genetic structure, which is the TGF-beta receptor 2 knockout. So this is really interesting that we see this. Why isn't the SOX1 tumor getting eliminated? It's full of T cells. And I told you before, those tumors should do better. They have better prognosis. They have better response to immunotherapy. Why does this tumor, we have almost as many T cells as we do cancer cells, and yet it's not eliminating? Well, it turns out that SOX1 is both a positive and it's a negative regulator of interferon gamma signaling, and interferon gamma signaling has both positive and negative regulators of immunity. So what's happening here is that you're getting upregulation of class 1, you're getting upregulation of CXCL9, recruiting a lot of T cells, but then PD-L1 also gets upregulated by having heightened interferon gamma signaling through the SOX1 knockout. So we made a prediction. This allowed us to make a prediction, which is that this tumor would be more responsive to immunotherapy. So this is a mouse. We took the mouse and we injected with a 1-to-1 mix of control and SOX1 knockout tumors. And just like we saw in perturbed map, if you measured what % of the tumors in the lungs were SOX1 knockout, it was 70%. They outgrew the control tumors, just like we saw with perturbed map. But then, as soon as you give anti-PD-L1, you see a flip. So now only 20% of the tumor burden is SOX1 knockout, because these tumors that were hyperinflamed are, in fact, more responsive to anti-PD-L1, just like we see in patients. And in fact, actually, JAK inhibitors have moved to being used now. There was 2 Nature papers to being moved as kind of a way to enhance potentially immunotherapy, and it's for this potential reason that you may be able to increase or actually decrease some of these negative checkpoints that are happening when you have heightened interferon gamma, especially chronic. Now, the other thing that we can do with the technology is ask additional mechanistic questions, and we can do that through spatial transcriptomics. So we can cut a layer of the tissue, not only measure by the protein barcode technology, but by, this is Visium SD, and you can see here that each of these clusters has a different knockout. This is actually unbiased clustering, so it actually found that there was TGF-beta receptor 2 knockout here and here without us even telling it, but found the same TME between those different knockouts. And when we looked, so this is a tumor, these are tumors that have the receptor for TGF-beta knocked out. When we look in those tumors, what is the most upregulated pathway? Well, it turns out it was the TGF-beta receptive, or TGF-beta pathway. Receptor gone, and yet the most upregulated genes were TGF-beta responsive genes. That was so weird to us, we thought we must have created, some sort of hyperactivating mutation. So we took those cancer cells, treated them with TGF-beta, and you can see that they are really a bona fide knockout because you don't have an upregulation of phospho-SMAD. Really a knockout. So what was happening there? And what it turned out is that these genes that are being upregulated in the receptor knockout tumors are TGF-beta responsive genes in fibroblasts. So why is that? Why is it that when we knock out the receptor, we get an upregulation of TGF-beta responsive genes in these other cell types? And I think the answer, or we think the answer from some other work we have done, is because even though we don't change the concentration of TGF-beta, there's no upregulation of TGF-beta in these tumors, we've removed the receptor from the most prominent cell type in the tumor, the cancer cells. So TGF-beta is sitting there latent. When it gets released from its latency, it no longer has a receptor to bind on the cancer cells. Where does it go and bind? It binds to the fibroblasts, and it binds to T cells. And so you have this receptor competition going on that's lost when you have the downregulation of the receptor that now causes the TGF-beta to act on the fibroblasts. And that is why we're seeing an upregulation of TGF-beta responsive genes. And what happens then is once those CAFs get activated, they exclude the T cells from the tumor microenvironment. And this is one of the reasons why we think you see a lot of TGF-beta receptor downregulation in colder tumors. And I think this is overall an interesting area that I'm going to come back to, which is how concentration dependent these relationships are. And this is really, again, about this spatial localization that is very, very hard for us to understand when we model, but it's a fact at play all the time. And this is something that we know as well is happening with T cells and how Tregs work, which is competition. They use the IL-2 receptor to outcompete IL-2 from effector T cells. Okay. This is just to show you, this is actually a piece of unpublished data. For those of you using Xenium, we can actually pair the technology with Xenium. So we can take these tumors, we run a Xenium run. This is Leiden clustering. And then you just take the slide after, you do multiplex imaging. So all you're doing is 2 rounds of antibody staining. And now we've resolved in this exact same tissue section, we can resolve, in this particular case, 20 different knockouts. This is an example of a JAG1 knockout, and now we can overlay the spatial transcriptomics on this as well using the Xenium. Okay, so in the last little bit of the last half of this talk, I'm going to tell you about a new story that we just had accepted, but is still unpublished. And we're moving to, we've been looking at ovarian cancer, which is one of, or it's the most deadly gynecological malignancy. 50% of women who develop ovarian cancer will die within 5 years because the treatments are very poor. And one of the hallmarks, so it's not a cancer that has a lot of mutations like something like lung cancer or melanoma. It has an unstable genome. So what you get with ovarian cancer is more like amplifications and deletions of genes and pieces of chromosomes are more common. So it's a very heterogeneous tumor in both its genetics, but also its tumor microenvironment. So this is an example of different tumor lesions from different patients. And you can see, actually, even if you take the same patient, look at different lesions, you will see very different tumor microenvironments. And we believe, although it's not been proven, that that's related to the underlying genetics. All right. So we wanted to model this. This is something that doesn't get done a lot. And I can't stress enough the importance that, you know, we're always creating these mouse models that are homogeneous, which is great because we like reductionist systems, but it doesn't model what's happening in a patient. So we thought, let's use the screening technology, functional genomics technology, to sort of model tumor heterogeneity. So what we did in this particular case, sorry, my headset may be falling off, And what we did in this case is we did a receptor-ligand, we did a receptor-ligand pair analysis from single-cell data, where we asked, okay, let's find genes that are expressed by cancer cells where there's a corresponding receptor on macrophages, and then ask, what are those particular genes doing to the tumor microenvironment? And these are the set of genes. There's, in this particular case, there's 35. Sometimes we go up to 100, but we try and stay low and focused. So none of these genes, when we knock them out, do anything to the cancer cells in vitro in either mouse or human cells. This is DepMap data. If you look, this is 17 different human ovarian cancer lines. You knock out any of these genes, and they don't do anything in terms of fitness to the cells. And that's not surprising, because most of these, or many of them, are secreted factors. So we asked, okay, well, will they do anything in vivo and in response to anti-PD-1? So this is what it looks like. We inject, in this particular case, we injected 8 mice. There was 5,000 distinct tumor lesions with all the different gene knockouts. We can map them, and you can look at them. And then you can ask the question, which genes influence tumor fitness, okay, before we get to the anti-PD-1, before we get to tumor microenvironment. And this is an example of what it looks like. So the genes that I want to point out here were really interesting to us because we actually didn't expect to see these genes come up. We see a lot of chemokines, like CCL7 and CCL3. We see Plower, for example, which is the UPAR, which is now considered a hot target on ovarian cancer, and we see MIF. And the reason why this was surprising to us, and should maybe even surprise everyone here, is we've knocked out this secreted factor. But I showed you in this previous slide, and I want you to remember this, because this is what our patient tumors look like, and it's what our model's tumors look like. Why is, if we lose CCL7 from this clone here, why aren't the neighboring clones producing it for it? Why aren't the T cells, even if they weren't recruited here, why don't they just move to this other tumor? And I think that's all about how these neighborhoods are working. And it's an important theme. It's really about this theme of the meeting, which is the ecology of the cancer and immune cell interactions. And so we decided, okay, well, is this real? Okay, we see it in our perturbed map. Can we recapitulate it? So we take a 1-to-1 or an 80-20 mix of the cancer cells to CCL7 knockout, and we completely recapitulate that. Even if we put 80% of the cells are the control, 20% are the CCL7. So you have an overwhelming amount of cancer cells that can still produce CCL7, but it doesn't matter. The CCL7 knockout still wins the race. It still outcompetes those other tumors. So what's happening there? Well, we did this technology, cyclic immunofluorescence, with our collaborators Sandra Santagata and Peter Sorger at Harvard, so we can measure, it's actually a more than 20 marker panel. And what we see in the CCL7 knockout tumors is a depletion of almost every immune cell type, including even endothelial cells. And this is what the tumors look like. And I don't know how well it shows up, but you can see you have dendritic cells and macrophages and T cells, and it's really quite cold here. And now when you look side by side, this is where, like, again, going back to that SOX1 TGF-beta receptor, but it's amazing in this particular case, to me at least, that we don't see mixing of these tumor microenvironments. And even more, that the one is giving it, we haven't changed any antigen, we haven't changed the cell's proliferation. All we've changed is this extracellular factor that's changing the microenvironment, and that's sufficient for the tumors to have a selected advantage to outgrow. So in white is the wild type, in green is the CCL7 knockout, and I think hopefully it's obvious to you, you can see that even when they're even sharing macrophages, and yet they have a different tumor microenvironment, even by eye. You can see even here, there's the knockout and the other tumor, and we can actually measure that, and what we're changing is the distance between the cancer cells and the ability of these T cells and dendritic cells to get near those cancer cells. And that's what it looks like here. So it's not just that the T cells kind of make their own way, or the dendritic cells make their own way. You also have the cancer cells which are pulling them in. And this is important. And actually, we went and stained. So in red is the control, in green is a CCL7 knockout. And now we've stained for CCL7 protein. And you can see how well it actually is being retained in these tumors. And a second question that came up, so you can see it's really locally concentrating there in order to provide that gradient for the T cells and other immune cells to come in. And again, even when you're side by side, so when we look, and we do it all the time, when we look at patient tumors, we will routinely see two different TMEs side by side. I don't know that this is all about CCL7, but certainly this is a factor. And so then we ask, why, which was a bit of a mystery to us, why aren't fibroblasts, endothelial cells, monocytes, macrophages producing the factor? Well, we think there's probably a feed-forward loop for some of these factors, and I think that is a common theme, that there's a feed-forward and feedback of cytokines and chemokines, and that's what we're seeing here. So when we did single-cell seq of these different tumors and looked at the macrophage population, one of the things that we see prominently downregulated with the CCL7 knockout in the cancer cells is that we see a reduction of CCL7 and CCL2 produced by the monocytes and macrophages, telling us that there's a feed forward where the cancer cells provide the factor, recruit these other cell types, which then can amplify the signal. Okay, and this is just to show you that when we look in human data, actually in human ovarian cancer, there's quite a good correlation between high and low CCL7 levels, and given how difficult it is to find or analyze RNA expression of these fairly low expressed chemokines, and it's a gene that's lost. And actually, even when we look at TIDE and TCGA data, you can see that there's a correlation between CCL7 and immune infiltration. Okay, so in the last part of the talk, I'll tell you the second half of that story, which is that we took the exact same library, put it into these mice, we put it into the peritoneal cavity, disseminates all throughout. These are modeling essentially metastatic ovarian cancer, where you get these clones everywhere, and now we treat with anti-PD-1. And here what we saw CCL7 driving enrichment, so those tumors became less sensitive to immune checkpoint. But this gene popped out at us, which was IL-4, which was really a surprise, and I'm going to tell you why it was a surprise to us and to the community, which is when we knocked it out, those tumors became more responsive. And I'm showing you this wheel here. This is the tumor size. This actually represents the relative fraction of tumors in the peritoneum of the mice with each of these different knockouts. So you can see here that only a small fraction of all the tumors actually carried an IL-4 knockout, and yet when we give anti-PD-1, you can see those tumors shrank more than any other tumor type. Now, why am I showing you like this? Because it's important to realize that these other cell types, every other color here, can still produce IL-4, and yet they're not compensating because it's required very locally, and I'm going to show you that in a second. But it's really quite remarkable how these clones are competing, and we're not changing anything cell intrinsically, we're just changing the tumor microenvironment through an extracellular regulator. So just to show you, we then created single knockouts where we knock out IL-4 from the cancer cells. If you do that, this is the control in blue. Nothing, this is their time to ascites, which is basically like survival. If you give these mice anti-PD-1, they don't respond, just like humans. If you knock out IL-4, nothing happens. They develop ascites just like the other mice do, and that was seen with perturbed MAP because we didn't see IL-4 come up when we didn't give anti-PD-1. But now when you have IL-4 knockout from the cancer cells and give anti-PD-1, those mice survive for longer. So why is this a surprise? Because everyone in the room has heard of IL-4. But what surprises us is no one, and I didn't know this until we started the work, is that people don't know where IL-4 comes from in a tumor. And that surprised us a lot, but it turns out that IL-4 is like a ghost. It's very, very hard to detect. This is single-cell data. This is IL-4. Looks like no cell type expresses it. This is all the time you see this. You can take an inflamed eczema lesion, and you will have a tough time finding IL-4. It is like the Kaiser Soze of tumors. It is a ghost, okay? And so we were like, when I first saw this, I'm like, well, this can't, there must be a mistake. We see that when we knock out IL-4 in the cancer cells, how can this be? Well, it turns out it's very hard to detect. And the other thing is, when you go and look at single-cell data, and I want to highlight this point, you'll see all these pathways come up, and you'll always see gamma, and you'll always see alpha. And that is true, that they are in play there. But we should not trick ourselves to think that other things are not at play. We should realize the limitations of our database. So what do I mean by that? Well, that output, when you say there's an interferon gamma signature, you're saying that because there's a Go term that has listed all those genes. But what if you don't have an IL-4 signature? What if you don't have an IL-34 signature? Well, then you're and you can't detect the cytokine, then you actually don't know if it's at play. And that's what's happening with IL-4. We don't have good IL-4 signatures. And there has been both pro and anti-tumoral roles, including two recent papers in Nature. So this is work that I did, was really led by Barbara Mayer, but my close collaborator, Miriam Murad. This is work we had done a few years ago, and actually comes from even earlier work we did when Julie Helft was still in Miriam's lab on migratory DCs. But we had found these population of dendritic cells in human lung tumors that had what looked like an IL-4 signature. Again, this is the extent of the IL-4 signature we had, not a very elaborate signature. And then when Barbara gave anti-IL-4, we saw this response in the lungs of the mice. And then in a follow-up paper, which was just published last January in Nature, Nelson Lamarche did some work where he looked for the IL-4. And I told you before, hard to find this gene. When he looked in the lung tumors, he doesn't find any cells expressing IL-4. So where is it coming from? Well, it turns out in lung cancer, and this is, I think, one of the themes that was mentioned before, which is what's happening is there's some signal coming from the tumor that's causing IL-4 to be upregulated in eosinophils and basophils in the bone marrow. And they are acting on myeloid progenitors so that when they get to the tumor, they're programmed. And so when Nelson depleted basophils, he actually sees something akin to what Barbara saw when she treats with anti-IL-4. So in this particular case, the tumor has found a way to get IL-4 through a signaling mechanism to the bone marrow, not through expressing it in the tumor itself. But in our particular case, we were seeing that when we knocked out the IL-4 gene, we saw this functional difference. That told us IL-4 had to be doing something, had to be coming from the cancer cells, right? So can't detect it by transcript, but at least to me, surprisingly, we can detect it by protein. Now, I think that we don't detect it by transcript because this is a very, very tiny population. It's about 3% of the cells. We confirm that it's really expressed because we actually knocked in TD tomato to IL-4 locus, and we recapitulate. And I want to mention something that when we knock in TD tomato to the IL-4 locus, we see in vitro maybe 4%, 5% of the cancer cells express IL-4, which is probably why it's so hard to detect. We can enrich those to 70%, inject them, it still goes back down to IL-4, even though each tumor is being started by a single clone. So there's something dynamically regulating IL-4 that the tumor wants it to be very low expressed, but expressed just in pockets of the tumor, and that's what you're seeing here. This is human tumor. We got patient slides. So in most patients, this is amazing because people say, oh, are mice and human the same? Well, in humans, we see almost the exact same frequency of IL-4 expressing cells, which is about 4% of cells in the tumor, and most of them are cancer cells. This patient was an outlier. Now, we took some spatial transcriptomics data, and you can actually see in this column here, IL-4, and it is in fact, you can see some from the myeloid, some from the fibroblasts, but if you look, and this is where spatial becomes important, very few of these are interacting with the tumor. So most of the tumor, within the tumor, most of the IL-4 is coming from the cancer cells. Now, IL-4 has never been described, has not really been described to be expressed by non-immune cells. It's mostly expressed by T cells, ILCs, basophils, eosinophils, which means that we don't know the transcription factors that drive its regulation. So we started to look at this. We made some predictions based on what's known about what controls IL-4 in T cells, the most famous being GATA3, which is absolutely required. We took these human ovarian cancer cells, which do express IL-4, and we knock out all of these different transcription factors shown to control IL-4 in T cells. Most of them did nothing. They don't change IL-4 levels. The white means IL-4 is still expressed. But when you knocked out IL-4, gone, IL-4, that makes sense. When you knock out GATA6, not GATA3, IL-4 is completely gone from these cells, and we can no longer detect it by ELISA protein production. So in cancer cells, the GATA6, in ovarian cancer cells, GATA6 seems to be the driver. Okay, so what is IL-4 doing? And so we went back, used cyclic immunofluorescence on those tumors to ask this question, and I'll show you here. So the first thing we see is in these tumors where IL-4 knocked out of the cancer cells just 4% of the cancer cells are expressing at any given time, and yet we see, even though there was no phenotype, remember, before we give anti-PD-1, there's no change in the fitness, but yet if you looked at the TME of these tumors, they look very different. There's a decrease in macrophages and an increase in exhausted T cells, which was telling us something extremely interesting, that even though you weren't getting tumor clearance, the T cells were now allowed in as the macrophage populations dropped. Now, when you give anti-PD-1, you had all these exhausted T cells here. When we just had IL-4 knockout, no effect. But when you give anti-PD-1 and those tumors shrank, what lights up? Well, it's the granzyme positive, perforin positive T cells, telling us that those T cells that had got in, they became exhausted, but now we could turn them on and have them start to clear those cancer cells. And this is just to show you the other thing that we see quite profoundly is an increase in these TLS-like structures within these tumors. This is an example when you have IL-4 knockouts, they're much bigger. You can see that here in this graph, telling us that not only were we getting more T cells in, but we were getting more TLS in these tumors, which is associated with better prognosis. So what happens in human? I told you there's not a good IL-4 signature, so if you go pull up all your single cell papers, you won't see IL-4 generally listed. So we've been trying to build one of these signatures, not easy to do. There's many reasons for that, and I think the community needs to work harder on this. But we have built 2 different IL-4 signatures, and when we project that, this is a very large data set, single cell data from human ovarian tumors. When we project that onto these patient tumors, where is it lighting up? It's specifically on the myeloid cells, not on the T cells, not on other populations. So really we see this IL-4 induced signature. And when we looked a little closer, it's really these Marco macrophages that are lighting up the IL-4 signature the most. So it looked to us that in patient tumors, the IL-4 is acting on the myeloid cells and most prominently on the Marco macrophages. So then we went back and said, okay, well, is that what's happening in these tumors? We've only knocked out IL-4 in the cancer cells. What happens? What is the mechanism driving this? Is it IL-4 on T cells? Is it on? So long story short, we knock out the IL-4 receptor now on myeloid progenitors using a mouse that Florent Genoux had developed, the MS4A3 Cree, crossed to the IL-4 receptor flocks. We now recapitulate. This completely phenocopies what we saw before. So what you're seeing here is wild-type ascites develop it in about 25 days. If you give anti-PD-1, these tumors don't respond. If you just knock out the IL-4 receptor on macrophages, it doesn't have any effect, just like knocking out IL-4 in the cancer cells. But now, when you knock out the receptor on macrophages and give anti-PD-1, you see a bona fide synergy between the two, and some of the mice never develop tumors. Actually, we have even better graphs, but this is probably the experiment that looked less crazy than all the others because the results were quite impressive. So then what we did is we did single cell analysis, and we asked, okay, well, what's happening here? So if we look at these data sets, this is where most of the controls are. They're actually, sorry, because let's see if the pointer, maybe you can't see it. No, I think it's not working. Anyway, what you can see, I'm going to skip it forward, is that when we knocked out the IL-4 receptor, actually when we knock out IL-4 from the cancer cells, or we knock out the IL-4 receptor from myeloid progenitors, what we see is the population that decreases. I showed you before macrophages decrease. It's very specifically the Selinap and Marco macrophages, which is the same population that in human ovarian cancer patients was displaying that IL-4 signature. So we think it's really a loss of this particular population. And what we're seeing here is a decrease in many of these genes, and actually Florent pointed out that some of these genes converge on particular pathways that he's very interested in related to iron metabolism and some other factors. Okay, and then I just have 3 more slides. So this is just asking the question, can we actually potentially translate this? The answer looks to be yes. So this is an example. If you give these mice anti-PD-1, you have no effect. But then if you give anti-PD-1 plus IL-4 receptor blockade, these are 2 drugs, we can now extend the life of these mice. And these are very aggressive tumors. We are talking about metastatic tumors we start treatment just maybe a week before they're about to develop ascites and close to die, and we're able to prolong their life for several weeks. So we are excited about that. And the other reason why we're excited is because after Barbara got these results, Miriam convinced our close collaborator, Tom Moran, who's a medical oncologist at Mount Sinai, to start a clinical trial. So he did. He took these patients who have lung cancer that have failed anti-PD-1. Okay, so they've been on anti-PD-1, some of them on for weeks and weeks and weeks. Nothing happened, the tumors didn't shrink. Takes this drug, so the nice thing about IL-4 is that there is an approved drug in Europe and in America. It's used all the time, it's extremely safe. It's IL-4 receptor blockade, it's used for eczema, they use it in children. People take it for years at a time with no negative effect. And when you do that, he took patients who had no response to anti-PD-1. All he did was add anti-IL-4 blockade, and some of these patients had almost complete responses and stable disease for several years even in some cases. And you can see that here in this woman's scans. You can see that the tumors, even after, this is actually old data, even after 1 year, her tumors didn't return. So extremely exciting because we think that, in terms of drugging, a pathway that we can drug, IL-4 is an excellent one because there's already a drug approved, and we can turn tumors that may be non-responsive to PD-1 to potentially responsive. Okay, so I want to sum up, and I think lots of people, this is probably not a crowd that I have to convince of the importance of neighborhoods, but some crowds you do. So in tumor biology, it was actually, I think Anna had mentioned it, which is, the kind of thinking in the field for many years is always cell intrinsic, and people like Nina and many other people have said TME is also important. Cancer guys ignore that, but even so, we still tend to think that cancer is driven by cell autonomous, driven events. So KRAS, P53, but even when we move to the immune space, we've tended to think it's cell autonomous, downregulation of class 1, upregulation of PD-L1. We think that that is happening, but there's also a dynamic regulation that, again, I don't have to convince this group of, which is when T cells or macrophages come in, they turn on programs that don't have to be genetically hardwired, that change the microenvironment. And the key of what I hope to show you today is that even in these mixed environments, it's enough that a cancer cell can program the macrophage that's just sitting beside it, and that over time, that is enough to give that tumor, that clone, a selective advantage. And this is where some of that tumor heterogeneity and tumor microenvironment heterogeneity is coming from, which is these events that happen where the tumor, a particular cancer cell, may protect itself by changing the macrophages, changing NK cells, changing fibroblasts around it, but not in a way to say that we need the whole tumor. It can be a 10-cell radius that results in this selective advantage. In this particular case, we see no selective advantage when we knock out IL-4 alone, but really as soon as we give anti-PD-1, we start to see it. And I think a key factor here that we have to think about, and it's very hard to model. It's why, this is no negative on anyone's models, but when we talk about organoids or we talk about other, very hard to model things like the concentration dependence, because we don't, as biologists, still have a good understanding of how dynamic it is and how local these events are. But this is something that, at least from this work and from other works, we're clearly seeing they're local, they're not moving far in radius, they're probably captured by receptors, so that's changing. And then the levels of expression are so important, and it's very hard for us to model that, even with our single-cell data, where we're actually not getting real single-cell data, but we're actually compiling lots of data together to composite a cell, but it doesn't tell us what's happening at a very local level, and this is something we have to understand. Also, that within these niches and these neighborhoods, there's a lot of feedback and feed-forward mechanisms where a cytokine may push its expression higher or control its expression, and then there is counterbalancing signals both from different cytokines, but also different cytokines doing two different things on two different cell types. So this is all things that we have to think about when we think of these local environments. So this is the last slide, just to say, if anyone's interested in the technology, you can either email me for the pro codes, or you can buy them from AdGene. We have a lot, really every tumor model we've ever tried it in works well, lung, ovarian, breast mets, pancreatic. And you can actually see, for those who do pancreas, this is a really cool example where you can see early on there's really a lot of mixing. We sort of thought that clonality was because the tumors were actually quite separate, but in reality, that separation, at least in this pancreatic model, comes much later as the tumors grow out, and you can see that here, that there's clonal dominance even when you don't see that early on. All right, and with that, I want to thank, again, the organizers, because I'm excited to be here at this amazing meeting, but the people who did all the work. The whole study, second half, all the ovarian, was led by Alessia Baccarini, who's a very talented assistant professor in my group, and Gurkan Mulliglu, who was a postdoc and is now an assistant professor at University of Alabama, and then all the other people in my lab who contributed, and then especially my close collaborator, Miriam Murad, Matthew Park, Nelson, Tom, and our good collaborators at Harvard, Sandro Santagata, Peter Sorger, and then all of our funding agencies for all the support. And thank you again. Happy to take questions.